Now, can you give us a quick update on this study, how it all started, and how far researchers are at this point?
Sure, yes. So this is a study that we undertook to try to prepare for the possibility of a future pandemic originating from viruses that may be hosted by bats. And so currently we have collected hundreds of samples from the ROM's tissue collection, and we are beginning to screen them for coronaviruses. We're also beginning to do our initial work to prepare our vaccine bank using the viruses that have already been described, so sequences of viruses that have already been published.
And what's really interesting is that the ROM has all these frozen bat samples. Why does this museum have all these bat samples on hand?
Yeah, so over the last couple of decades, they have gone all around the world collecting these bats, and in their case, it's largely for the purposes of understanding bat taxonomy, and so how different bat species relate to each other, what the differences or similarities are in their biology. So that's the main reason they have it, but it's also a great resource for us then as well.
And I'm going to ask the question that nobody really wants to hear right now, but how likely are we to see future pandemics similar to the COVID-19 pandemic?
Well, I'll sort of answer that in two ways. I think if the question is how likely are we to see transfer of coronaviruses into the human population in the future, I think that is very likely. As you mentioned, we've seen it now 3 times in under 20 years with SARS, MERS, and COVID-19. So I think that's very likely, and that's why we're doing this study. Whether that will result in a pandemic is, I won't really speculate on, because that really depends how we respond to that outbreak or that incidence.
And what is it about bats in particular that makes them connected to these epidemics, SARS, MERS, and COVID-19?
So the connection there is that in each case, there are viruses that are closely related to the virus that crossed over into humans found in bats. So it's not necessarily that the virus went directly from bats to humans. There may have been other animals involved, but bats appear to be particularly important reservoirs for a broad diversity of coronaviruses. And so we think that's an appropriate place to look if you're looking for what coronavirus might cause the next outbreak in humans.
All right, Dr. Troyer, we've got a minute and a half left, but how many vaccines is the team looking to make? The ROM has over 1,000 bat samples, so what is the target here?
So our target would be likely in the hundreds. So, the idea there is that if we can have something that's fairly closely matched to a number of viruses, to use something right at the beginning of an outbreak, it may not have to be an exact, complete match. We're seeing now, for instance, in the current outbreak, that the vaccines that we're using are effective even against the coronavirus variants that have emerged. So similarly, in our strategy, it doesn't always have to be a perfect match, but we'd like to really cover the diversity of possible coronaviruses that might enter the human population.
All right, just about 25 seconds left, doctor, but considering the timeline it usually takes to produce a functional vaccine, how long would it take to develop this vaccine bank?
So we can begin fairly rapidly. We here at Western, there's another researcher that we work with, Dr. Young Kang, that has a system for a SARS-CoV-2 vaccine that I'm also working with, and using their technology, we can begin on that essentially immediately. And this would be something that's ongoing. We'd hope to have a lot of this in the next year or two. But then we'd hopefully be able to continue this project into the future so that as we find new coronaviruses, we can incorporate those into the bank as well.